The properties of the epigenetic clock are visualized in these two graphs. On the right hand, you see the Vitruvian man that depicts, of course, the perfect physical dimensions. However, for the longest time, we didn't have a time axis. We didn't know what is the perfect age of the liver in a 40-year-old, what is the perfect biologic age of the lung tissue, for example. And the epigenetic clock is a biomarker that allows you to assess the ages of every cell type, every tissue, and every organ in the human body. And to tell you right away, in a perfectly healthy person, all body parts have the same age, with the exception perhaps of female breast tissue that seems to age faster. But in principle, then, the output of the clock would be, for a 40-year-old man, it would say that his heart is 41, his liver is 38, his bones have the age of 43, and so you get numbers. When you get diseased, there will be deviations, you know. So it could be, if you're obese, your liver will be older than the rest of the body. But stresses act in a tissue-specific manner, you know. The left panel visualizes another property of the epigenetic clock. You notice there's a baby and a very old man. And the epigenetic clock really applies to the whole age spectrum. It applies to prenatal brain samples. It can be used to study development. But of course, here our focus is in aging, you know, and it applies to all the way up to supercentenarians. So the epigenetic clock is based on an epigenetic measurement, which is known as DNA methylation. And cytosines of the DNA sometimes get methylated. And that's what it's based on. So the epigenetic clock applies to methylation data that can be measured using the Illumina array. I do want to say the epigenetic clock is really a platform independent. You can pretty much use any method to measure methylation, but I usually use these three platforms. The oldest Illumina chip was known as the 27K array. Then most of us have the 450K data, which measure 500,000 cytosines in the genome. And now the most recent version is called the EPIC chip, which measures over 850,000 locations on the DNA. And each location is known as a CPG, which is a dinucleotide. And the measurement gives you a number between 0 and 1. 0 means this cytosine is unmethylated in all cells. 1 means it's methylated in all cells. And let's say 0.5 would say that in 50% of chromosomes there is a methyl group, and in 50% there isn't at this particular location. So I talk about an aging clock, epigenetic clock, and I've been criticized for this unscientific use, but therefore I felt I should give a definition of what I call an aging clock. So in my opinion, the word clock implies that you have a very, very accurate aging measure. For example, if you measure the amount of gray hair, that's not accurate enough for measuring the age, you know. And so accurate is defined here as a very high correlation, bigger than 0.8, between the estimate of the age and the chronologic age of the subject in subjects that are aged between 0 and 100. So in other words, imagine you have a new method for measuring age, or let it be telomere length, and you have 1,000 people. They're aged between 0 and all the way to 100. And you now assess telomere length in these people and correlate it with chronologic age. If that correlation is bigger than 0.8, I would say you're dealing with an aging clock, according to my personal definition here, you know. And in any event, so there are many candidate aging clocks that you can think of. I mentioned telomere length. Some people look at gene expression levels. A famous one is P21 expression, protein expression, protein levels, and of course methylation. And the thing is, at this point, I'm not aware of any data type that gives rise to a very accurate aging clock except for methylation. Turns out, once you have methylation data, it's very easy to define an aging clock. So the work I described today was published less than 3 years ago, where I developed a multi-tissue biomarker of aging. And to develop that clock, I downloaded publicly available DNA methylation data. So fortunately, in our research community, people have the good habit of posting their data after their paper is accepted, and many people include relevant clinical covariates such as age. And so, in other words, the data were readily available. And so, statistically speaking, the analysis is straightforward. You have an outcome measure, chronologic age. You have covariates, these CPGs, and you have a regression task. And so I used a so-called elastic net regression model, which automatically selected the CPGs that best predict chronologic age. And there were 353 CPGs. 193 of these CPGs tend to get hypermethylated with age, and 160 lose methylation as we age. But I want to tell you right away, when you look at these individual CPGs, most of them have a very weak correlation with age, So we have here a situation where the total is definitely more than the sum of its parts. Although individually these CPGs have small correlations with age, in combination they have a very strong correlation. So the epigenetic clock method is comprised of 3 steps. In step number 1, we measure these methylation levels of these 353 CPGs. And so we have 353 numbers. And then we form a weighted average based on the regression coefficients. And that weighted average is not yet in units of years. So therefore, in step 3, we transform this average so that it's in units of years. And the result is known as epigenetic age or methylation age. Now, what is so surprising about the method is that I use here the identical algorithm for every tissue and cell type. Notice in step 1 you do not input tissue type. You don't say you're dealing with T cells or neurons or hepatocytes. It's just any source of DNA. I should say with the exception of sperm. The clock doesn't work in sperm-derived DNA. So here I show you how accurate the age prediction is. If you look at the upper left panel, you notice that in these completely independent test data, we achieve a correlation of 0.96 between the age estimate and the chronologic age. The accuracy is so high that well-respected aging researchers rejected the method because they felt if something is so accurate, it cannot possibly measure biologic age. But let's look carefully why it is so accurate. It's so accurate because we are dealing here with tissue samples. On the y-axis, you see ages from 0 to 100. So if you have a broad age spectrum, that's when you see these very high correlations. If we analyzed middle-aged people, let's say between 40 and 60, the correlation would only be maybe 0.5. So the correlation obviously reflects the range. What is interesting, perhaps, for this audience is that the epigenetic clock works in sorted cells. So you could analyze CD4 positive T cells, or cytotoxic T cells, or monocytes, or B cells, any blood cell type, it works. It also works in PBMCs and whole blood. it also works in neurons and in glial cell, and of course in a host of other tissues that are shown in the different panels. So in certain ways, the most important question was whether the epigenetic clock measures biologic age. And in order to answer that, I felt the need to relate it to many aging phenotypes, and also there's now there's a growing field where people use this biomarker. And here I sort various conditions alphabetically, and these are conditions that are typically studied in aging research. So for one thing, yes, the epigenetic age is accelerated in Alzheimer's disease, as long as you look at the prefrontal cortex. Similarly, amyloid load is correlated with age acceleration. Body mass index has a strong relationship in liver, a much weaker relationship in blood. Calorie restriction, liver, cell passaging, cellular senescence, Hayflick limit, all of that relates. Interestingly, cognitive performance relates. So, I mean, it's unbelievable, but if you take the blood samples of a 70-year-old, and if the blood exhibits accelerated aging, their cognitive functioning scores tend to be lower. And the same holds also for brain tissue. HIV status has actually a very strong effect. That's why I will present that later. Huntington disease is associated with age acceleration. Menopausal. Interestingly, the clock predicts all-cause mortality. So a blood measure is predictive of how long you live. It's not very accurate, but it's statistically very significant. Gender relates. Men tend to be 1 or 2 years older than women, according to the epigenetic clock. Sleep has been implicated, and walking speed. So, because the previous talked about telomere length, I wanted to give you some results from the Framingham heart study, where Brian Chen and others looked at leukocyte telomere length in the upper panel, the y-axis, versus chronologic age. And what we see here is that there's a correlation of minus 0.27. And that's, of course, the well-known result that telomeres shorten as we age. Now, on the very same subjects, we also had methylation data available. And therefore, we could correlate methylation age on the y-axis versus chronologic age. And in the very same subjects, we now observe a correlation of 0.81, you know. And that's my one slide to promote the idea that aging researchers should really look at methylation, you know. Incidentally, I'm completely sold on the merits of telomere length, and these are complementary biomarkers. So here I just show you a couple of papers that have looked at the correlation between telomere length in blood and epigenetic age, or in adipose tissue. And the answer is there is no relationship, you know. So methylation age is an independent variable from telomere length. And just to be clear, of course, these two markers are confounded by chronologic age. So my statement is the following. If you have a birth cohort, every person is exactly 40 years old, and you measure their methylation age, and at the same time you measure their telomere length, and now you do a scatter plot, you will see no relationship, you know. And that's exciting because we now have two independent biomarkers of aging, you know. So let me show you some results in HIV. So Andy Levine and I looked at the effect of HIV infection on the epigenetic clock. And we analyzed many methylation data sets. Many of them deal with brain tissue, so 3 brain methylation data sets and 4 blood methylation data sets. And the brain tissue samples came from the National NeuroAIDS Tissue Consortium. And the brain, so most people were on average 71, at age at death was actually quite early, around 46 years. And here I show you how methylation age on the y-axis correlates to chronologic age in all brain samples, where we ignore brain region. And what we observe, red denotes HIV-positive brain samples and black HIV-negative brain samples. And what you see right away is that the red dots lie above the black dots. Just never mind the statistics, you know. And that, so what does that mean? The methylation age estimate is higher than that for controls. And when you do the math, the p-value is 10 to the minus 8, you know. So that suggests then that the brain samples from HIV-positive subjects unfortunately exhibit severe age acceleration. By the way, when I call something severe age acceleration, it means something like 5 years. For me, that's a strong effect, you know. And just another digression. If you ask me what condition has the strongest effect on age acceleration, it's not HIV, it's actually Down syndrome. That I find that has a very pronounced effect on age acceleration in brain and blood, you know. In any event, the problem with this all-brain analysis is that it's confounded by brain region, and therefore we carefully looked at individual brain regions, for example, the occipital lobe, and we find again a very striking effect. You know, the age acceleration in the occipital lobe is very pronounced. We find a weaker effect in the cerebellum, but interestingly, we find no effect whatsoever in the frontal lobe, you know, and I can't explain it, you know. Why would the frontal lobe not show age acceleration? From the same brains, we also had CSF-based estimates of viral load, and in certain ways it's interesting to ask, is viral load correlated with epigenetic age acceleration? And there was no relationship, unfortunately. I mean, I wanted, you noticed I talked about age acceleration, and I just want to give you an explanation of what is meant by that. So look at the scatter plot. What one can do is one can form a regression line through the dots. And if there's a brain sample that lies above the regression line, then it's older than expected, and then we say it has positive age acceleration. But there are some brain samples that lie below the regression line, and they have negative age acceleration. You know? And so this measure of age acceleration is, of course, plotted, for example, here on the y-axis, you know, and it can be positive or negative. Now, this was the first brain data set. We call it the discovery brain data, but then we generated a second brain data set, the validation brain data. And again, we found, which was much smaller, but again, we found that the cerebellum exhibited age acceleration. And again, the frontal lobe did not, you know, so there you have it. So I think it will be interesting to look at what brain regions show age acceleration and which don't. Moving on to blood tissue. There's no doubt blood tissue exhibits age acceleration in HIV-positive subjects. Here I show you 2 data sets, but there are many other published data by now that show the same effect, you know. Also, a question is whether this epigenetic age acceleration measure correlates with various blood cell types. And the answer is yes. You know? So for example, there is a negative correlation with the abundance of granulocytes. There's a negative correlation with the abundance of CD4 T cells. There's a stronger association with the abundance of naive CD8 T cells and also with exhausted or senescent T cells. And all of these results were obtained in HIV-positive subjects. You know. So stepping back and thinking about the model, in certain ways the question is, why do we observe age acceleration? You know? And that is, by the way, the greatest weakness of the epigenetic clock, that we don't completely understand the underlying biological mechanism. That's of course the great, the very attractive property of telomeres. There's a beautiful mechanism. We know all the enzymes that control telomere length. With epigenetic age, we are only at the beginning. We know a few genes that relate, but yep. In any event, so one plausible model would be that HIV might shorten telomere length, and telomere length then might lead to shortened telomeres might increase epigenetic aging. However, this model is, in my opinion, not plausible for many reasons, but one reason is that telomere length does not relate to age, right? Age acceleration, sorry. And so I don't believe this model. Model number 2 is plausible. So it would say that HIV leads to, for example, exhausted T cells or changes in blood cell composition, and they might drive age acceleration. And in blood, that is a plausible model. However, it's much harder to understand that model in brain tissue. Remember, that's where we also found an effect. So I feel this requires further study. Model number 3 is what we call the independent model, and that says that HIV accelerates epigenetic aging independently from its effect on various blood cell types. So these are just different markers of this HIV infection. And again, our data suggests that this could be a plausible model as well. So I want to talk briefly about another study, HAND, the so-called HIV-associated neurocognitive disorders. This is work done by Andrew Levine, Austin Quack, David Moore, Christian Akim, and many others. So here we looked at, again, epigenetic aging in the occipital cortex, and now we are only dealing with HIV-positive subjects, okay? So methylation age was again highly correlated with chronologic age, and now the coloring indicates HAND. So red means the person had HAND, black means the HIV-positive subject didn't have HAND. And interestingly, we do observe a hint. I call it marginally significant, p-value 0.03, You notice there's one severe outlier, so after we remove it and we focus on these subjects, the p-value is still around 0.03, So I feel it's a tentative finding. It certainly warrants additional studies whether, in certain ways, the epigenetic age acceleration in the brain, here occipital cortex, but maybe other brain regions, correlates with HAND status. So our next steps for this project are really to carry out an epigenetic profiling analysis of several human tissues. And so I'm very happy to collaborate with the NNAB team, Andrew Levine, Susan Morgello, Elise Singer, Jonathan Sett, and many others who have these very valuable tissue banks. And what we're doing now is we will look for epigenetic age acceleration effects in multiple organs, lung, kidney, liver, heart. Personally, I would like to profile 10 different areas of the body, And also, we want to understand which measures of tissue pathology correlate with epigenetic age acceleration. For example, what measures of liver damage or what measures of brain pathology, neuropathology correlate with age acceleration. And of course, the question is, what's the role of art? I should tell you, the epigenetic age acceleration can be observed in people who received art and also in people who before they received treatment. So it's not as simple as if you provide art that you reset the epigenetic aging. I think that's not the case. And of course, we want to look at which other so-called HANA conditions, HIV-associated non-AIDS conditions, relate to epigenetic age acceleration. So I want to shift gears and just provide you a broader view of the aging applications. The ultimate question is, does the epigenetic clock actually predict all-cause mortality? And why do we want to answer that question? Why we want to show that it measures biologic age as opposed to chronologic age. And so there were several papers out there that already showed that methylation age of blood predicts all-cause mortality. These are Ian Deary, Naomi Ray, Riccardo Marioni, and others, where we collaborated, and we analyzed 4 different cohorts to show that. Then there was a Danish study that showed it and a German study. I want to tell you, tomorrow a paper will come out where we carried out a very comprehensive meta-analysis. This was an effort led by Brian Chen, Luigi Ferrugi, and many others, where we analyzed 13 different cohorts, in total over 13,000 people. And from each person we had blood methylation data and follow-up data to know, well, let's say they were followed for 20 years. So we could assess whether the blood collected in the 1990s actually ended up predicting their lifespan. And here are the results. So these are so-called meta-analysis forest plots. These are the different cohorts. And focus on the p-values. Each of these forest plot corresponds to a different measure of age acceleration. There's something called, anyways, there's different ways of measuring age acceleration. But any measure we ever looked at leads to a hugely significant association. P-value 10 to the minus 11, 10 to the minus 9, 10 to the minus 43. You know? So yes, these data predict mortality. What is particularly striking is that these models predict, or epigenetic age acceleration predicts mortality even after you adjust for all the covariates and risk factors that you could think of. We certainly adjust always for chronologic age, that's a given, but also body mass index, education, alcohol consumption, smoking status, history of diabetes or cancer, hypertension, the amount of physical activity you do. You know, you adjust for all of it, and it still is a significant predictor of mortality. And that shows, in my opinion, that it relates to an innate molecular aging process, you know, that, yeah. I looked at centenarians, and it turns out if you have a parent who lived till age 100, then arguably you probably inherit longevity genes, and therefore you might expect that your epigenetic age in blood is younger than that of a person who doesn't have a centenarian parent, you know. And the answer is yes, that's true. So at least in Italians. So this was a collaboration with Claudio Franceschi, Paolo Garignani, and many others. I'm cautious about it. I emphasize Italians because I feel it needs to be validated in other populations, you know. I want to mention various other applications. Menopause. So we looked at menopause. This is a study with Morgan Levine and many others, where we actually showed that menopause accelerates epigenetic aging. And that's a causal claim. Notice the title is a strong claim, but I feel we're confident in making it. Let me describe the study. So we looked at epigenetic age acceleration vis-a-vis, so that was the outcome, epigenetic age acceleration. And for these women, we also knew the age at menopause, their smoking status, hormone replacement therapy status, age at menarche, and so on. And we looked at 3 large cohorts, WHI, and so on. We found a p-value 10 to the minus 4. So there is an association. It's a very weak association, though, right? You need a large sample size to find it. However, association is not causation. However, to make a causal statement, we made use of the fact that for some of these women, unfortunately, had undergone what is known as surgical menopause or bilateral oophorectomy. So for various reasons, the ovaries were removed, and that induced, of course, menopause. And for these women, we also measured the age acceleration, and sure enough, surgical menopause increased age acceleration. And if you make the assumption that surgical menopause is a quasi-random intervention, some women have simply bad luck, they need the procedures, others don't, then this is almost like a randomized trial, you know, with the big caveat that it may not be random. But also then we looked at, we made a genetic argument where we looked at SNPs, genetic markers that had been found to be associated with early menopause, and one of these SNPs also was associated with age acceleration. So that's what is known as a Mendelian randomization argument, you know. And finally, we looked at the effect of menopausal hormone therapy, and it turns out that women who had menopausal hormone therapy, their buccal cells, the cells inside the cheeks, they were significantly younger than those of women who didn't have hormone replacement therapy. And I should mention, we also looked at the effect of hormone therapy in blood, but we couldn't find an effect in blood, you know. I'm not sure how much time is left. Over? Okay, sorry, then I'll stop it. Let me just mention Hispanics age more slowly, and I'll stop with that. And I want to thank my collaborators. Thank you very much.